Hello and welcome. I'm Mark Lovern, Senior Vice President of Commercial and Product Strategy for Certara's SimSip business unit. And it's my pleasure today to speak with Ava Berglund, one of our senior clinical pharmacologists here at Certara. Particularly, we're going to speak about drug-drug interactions for CYP3A substrates and how PBPK can be of help to those programs. Ava, if you would maybe tell our viewers a little bit about your credentials in this area.
Thank you, Mark. So I'm Eva Berglund. I'm Senior Director in Clinical Pharmacology and Regulatory Strategy here at Certara. So I work as a consultant in drug development in the IDD, Integrated Drug Development of Certara. Before I joined Certara, I was a regulator. So I was a clinical pharmacology reviewer in the Swedish agency, and I've been deeply involved in guideline drafting, for example, DDI guidance and also PBPK guidance at EMA.
Great. So I've definitely come to the right person. So my question today, Ava, arose out of a video we recently put out around how PBPK can be used in some instances to make DDI label claims. And we got a lot of questions about that, including what human data do you typically need to make robust claims in this regard? So can you tell us a little bit about that?
Yeah, so to simulate up a DDI with a CYP3A inhibitor, so you would need to know the contribution of CYP3A to the drug elimination. And you would get that information either, and that's more common, from a DDI study with a strong CYP3A inhibitor. But you can also use a mass balance study if you have very clear results and also very good supportive in vitro data. So you would use that, and then you start to simulate up different DDI scenarios.
Okay, and so once you have that data, what sort of scenarios can you simulate and potentially get, label claims for?
So then you usually start with simulating up based on the DDI study or mass balance study, the effect of moderate CYP3A inhibitors. And usually there you want to actually simulate the range because you want to make a claim that works also for both the stronger and the weaker in that class of moderate inhibitors. So you start there, and then you might, if you need, you might simulate up the effect of a weak inhibitor. If you have a big CYP3A contribution, you simulate up then the effect of strong inducers, for example. And there's many situations here where you can use PBPK in order to translate this into labeling recommendations. And that is very much done in our labelings, both in EU and the US.
So it really sounds like if CYP3A is the primary metabolizing enzyme, that there's quite a bit that can be done with PBPK once you have the right data.
Yes, absolutely. CYP3A is really the enzyme we know most about. We have so much cases and so much experience when it comes to PBPK simulations in this respect, both drugs inhibiting CYP3A and inducing CYP3A. And this is very much translated into the labelings of today.
Thank you. That's really helpful, Ava. I really appreciate you taking the time to speak with me today. And I hope you have a great day.
Thank you too, Mark.